a shot, a jab, inoculation, immunization, a vaccine. Whatever you call them, these medical advancements have been instrumental in improving the quality of life and ensuring survival in some of the most vulnerable age groups of humans. Estimates are that vaccines alone prevent 2 to 3 million deaths every single year. But how do they do this? What's in a shot? Hello, my name is Shelby Bradford and I'm the Geeky Goth. Today, we're talking all about vaccines. What are they made of? And how do they work? First, a little bit about me. I got my bachelor's degree in immunology and microbiology. Then I decided to hang out for a little bit more science, and I'm currently a graduate student pursuing a PhD in these same fields. Along the way, I learned not only about immunology and microbiology, but also took coursework in genetics, virology, cancer cell biology, and vaccines themselves. Today, I'll be applying all of this background to help us get a sense for these questions. What are vaccines and how do they work? It's a big topic, so let's jump in. Let's start with what the vaccine is made up of. In the fluid in the syringe, the most important part is what we call the antigen. The antigen is the part of the vaccine that we're training our immune system to recognize. We'll talk about these in more detail in a moment. Some vaccines also include a substance called an adjuvant. These adjuvants have the job of kickstarting our immune systems in some vaccines, most often by mediating inflammation, a crucial process in our immune response. Anything and everything else in our vaccine, the water, the sugars, the salts, are called excipients. These actually have no active role in the vaccine itself. They're just there to keep everything else, our adjuvants, and most importantly our antigens, content when it's in the vial. Since almost all of our action then is on these antigens, let's look at them in more detail. Vaccine antigens come in a couple of different forms. These can be grouped into 5 broad categories: live attenuated, killed or inactivated, subunit, toxoid, and most recently, genomic. Let's go over the properties of each of these. Live attenuated vaccines are kind of exactly like they sound. They also tend to be the OG vaccines. So a lot of our earliest vaccines are live attenuated, whether that's smallpox or the vaccine against tuberculosis, BCG, and many other viral vaccines like mumps, measles, and rubella. The way these work is pretty straightforward. We have our actual pathogen, either a virus or, in some cases, a bacterium, and it can cause disease. What we do to make a live attenuated vaccine is we either take this actual pathogen and we culture or grow it in such a way that it weakens itself and can't infect us as well, or we take a similarly related organism and use this as the vaccine antigen instead of the disease-causing organism. So these vaccines are pretty straightforward. You attenuate or weaken an existing pathogenic organism so that it doesn't cause disease in people with healthy, intact immune systems. Now, as the name implies, the antigens in this vaccine are still capable of reproducing, making more of themselves. This has an advantage because these vaccines are plenty stimulatory on their own. We don't need an adjuvant. However, because these organisms can multiply, they are not recommended for individuals who have any compromised or suppressed immune systems, including pregnant people. Next up, killed or inactivated vaccine antigens. Similar to our live attenuated vaccines, the antigens here are using the whole organism. However, instead of just weakening or using a similarly related organism, here we've just killed, or to avoid that whole viruses are alive debacle, inactivate the pathogen of interest. Vaccines that fall into this category include hepatitis A, the shot version of polio, as well as the rabies vaccine. Let's explore what this looks like. If you were to be infected with one of these viruses normally, it's perfectly capable of reproducing in your cells. And that leads to your cells eventually dying, and you have this viral disease. In the case of these vaccines, this virus is inactivated, so it can't reproduce in your cells, but your cells will still respond to it as if it were the actual virus, and so they will destroy it and build immunity towards it. Because the antigens in these vaccines aren't capable of replicating, there's less of that contraindication that we saw with the live attenuated vaccine antigens. However, because these organisms aren't replicating and stimulating the immune system that way, we do need to use that adjuvant to really kickstart all of that activity. But because of the overall lower stimulation, we do see the need of boosters with many of these vaccines. So far, these vaccine antigens have involved the entire organism, be it alive but weakened or killed/inactivated. However, as science advanced and as vaccines evolved, we realized it could be advantageous to make vaccines that only used parts or subunits of these pathogens. So we can use specific features, be they proteins or sugars, from these organisms to train our immune systems to recognize them, and then by proxy be able to identify the whole pathogen if we were to be infected. As we can see in these two examples here, pathogens have a lot of stuff on them that make them up. If we were to get infected by any of these pathogens, our immune system would tailor its response to these proteins and sugars already. Using a lot of really cool science, researchers and scientists were able to purify these proteins and sugars that are a part of these pathogens, and we use them to generate our immune responses in the absence of the actual bug itself. These might involve structural proteins or proteins that are released to try to waylay our immune systems, or it might involve sugars that are a part of the protective layers of some bacteria. For many proteins, this is as easy as just purifying the protein you like, inactivating it if necessary so that it doesn't have any function in the body, and then letting it go, and our immune system does the rest. But for some of these antigens, like sugars, that can be a little bit trickier, scientists developed a way to make them good antigens by attaching them to inert molecules that we'll call linkers here. In this way, these sugars are actually recognized by the immune system so that when they come in on the bacteria itself, the immune system's actually going to be able to pick it up. Once they've done their job, the immune system and just our regular cells will be able to just destroy these proteins and sugars. Now, these vaccines have a lot of advantages. Because there's no organism whatsoever, they don't see the same sort of contraindications that can be had when you have definitely live organisms, but even sometimes killed or inactivated pathogens. This means that they can be given to almost anyone, and that's great because that's more people protected against disease. However, similar to what we saw with our killed and inactivated vaccine antigens, these subunit vaccines tend not to be as stimulatory on their own, and so they'll require adjuvants as well as booster shots to really build up and maintain that strong immune protection. The next type of vaccine antigen, those of toxoid antigens, are really like specialized subunit antigens. The reason these are really getting their own classification is here we're only directing our immune response to toxins that are produced by the pathogens. These are used when the entire disease is really driven by these toxins. Our immune systems can take care of the pathogens just fine, but the toxins they produce that can linger even after the organisms are dead are really more of the issue. So for these, scientists purify these toxins, which are often proteins themselves, but they inactivate them to produce toxoids. Like we can see depicted here, normally these produce toxins even in the absence of the pathogen, are able to kill or otherwise cripple our cells. In the case of these toxoids, they don't have any activity because they've been inactivated, but they look similar enough to the actual toxins that the immune system can learn to recognize them. Then, if an individual is infected with the pathogen and that toxin is produced, the immune response will be able to neutralize the toxin, preventing the disease. Our final type of vaccine antigen is actually very new to the playing field. However, vaccine antigens using genomic material have been studied for decades. As probably all of us have seen recently in the news, if we're infected with a virus that's not been seen before, the immune system is really going to take the hit in terms of cell damage and just overall health before an immune response can be mounted. The way that these first-generation genomic vaccines have been designed is a single piece of RNA encoding one protein is isolated and then packaged into this protective lipid layer. These lipids are analogous to the same lipids that make up our own cells. This viral RNA gets deposited into our cells, and our cells just simply follow the instructions and make the protein. Now, because this is a viral protein, our immune system immediately recognizes it as something that doesn't belong and mounts an immune response to the protein. Similar to what we saw with our subunit and toxoid vaccines, this means that if this protein comes in again, this time on the actual virus, the immune system will already have its game plan together and will deploy it on these proteins on the virus. If you want to see this in more detail, we talked about it in a previous video, and I encourage you to check it out. Now, these vaccines, even though they're new, we do have a really good idea about their profile and properties. For one thing, because this is literally just genomic material wrapped inside of some fatty membrane, these vaccines are incredibly safe. However, because of the nature of RNA, as well as the structure of these lipids, these vaccines are sufficiently stimulatory, and so they don't even need an adjuvant. However, one potential drawback to these vaccines is, again, because of the nature of RNA, they may require some pretty cold storage temperatures because RNA does tend to be pretty unstable. However, in terms of safety and effectiveness, these vaccines get a big check. A second style of these genomic antigen vaccines takes a different approach. These use DNA as well as what we'll see as a vector to actually deliver their antigen. As we've talked about a couple times by now, in a normal infection, a virus or other pathogen will infect us, and in the case of a virus, will rapidly take over, hijack our cells, make more of themselves before our immune system can even respond. However, similar to our RNA-based genomic vaccines, these DNA-based vector vaccines are going to provide our cells with the instructional material to make some of these kind of trademark proteins from the virus. Unlike our RNA vaccines, which we're using just these small bundles of lipid to move the RNA into our cells, these style of vaccines use what's called a viral vector, hence why they're sometimes called viral vectored vaccines. So, what's a viral vector? It is a virus, but these are viruses which, first of all, rarely cause severe disease in humans, if they even usually cause disease in humans. And most importantly, these viruses have had their genetic material snipped up to take out, at the very least, any genes that can make proteins that make us sick, and oftentimes genes that allow the virus to replicate itself. So these viral vectors are really just shuttles for the instructions we want them to move into our cells for us. So these viral vectors enter our cells because that's really all they know how to do, is try to get into a cell. However, again, their genetic material has been modified. And most importantly, in addition to snipping out the bad stuff, we also put in a small piece of DNA in this case that's going to have instructions for some of these specific proteins from a different virus that we're interested in. This DNA gets shuttled into the nucleus. Here, it's going to be read and turned into RNA, and then it follows the same process as we just talked about with our RNA-based vaccines, where it gets turned into a protein that gets recognized by our immune system as something that doesn't belong. Now, it is incredibly important to point out here that this DNA that gets introduced isn't capable of just inserting itself into our DNA. That takes a lot of extra proteins to do the work that we don't have and that this virus isn't bringing with it. However, even if somehow this DNA worms its way into our DNA in this cell, as soon as our immune system sees these unfamiliar proteins, this cell is going bye-bye. So it wouldn't really be sticking around with that viral DNA anyway. Now that we've introduced the main players in our vaccine, let's take a closer look at the characters of our immune system to see how protection from these vaccines is made. We talked in a previous video about how the first step in an immune response for either a vaccine or for an infection is to initiate inflammation. This inflammation is responsible for recruiting our immune cells. However, during a vaccination where there's very little threat to the body, many of these cells, like neutrophils, natural killer cells, and macrophages, are actually going to be on cleanup duty. They're going to take care of getting rid of any infected or damaged cells, or just any floating around particulate that might be from these infected or damaged cells, or from the antigen itself. Really, the MVP during a vaccine response is the dendritic cell. This cell is responsible for grabbing up this antigen and then moving through the body to the lymph node, along the way breaking that antigen into smaller pieces. Once it gets to the lymph node, a lot happens. Once our dendritic cell gets to the lymph node, it will have broken apart that antigen, either a protein or sugar from one of our subunit vaccines, or maybe a whole organism from our live attenuated or inactivated vaccines, into small pieces, which we also call antigens. And it's going to scout through immune cells that we call T cells in the lymph nodes. And these guys are pretty important. Let's look at why. So the dendritic cell is going to show off some of the pieces of this antigen, smaller antigens now, to some of these T cells. And this is going to activate these T cells. And some of them will become killer T cells. And their job is going to be to return to the site where this immune response is ongoing with all of that antigen and inflammation. And we'll see what it does when it gets there, although you could probably guess by the name. But you're probably asking yourself, well, where do B cells come in? That's what we always hear about in terms of vaccines, B cells and antibodies. That comes into play with some other types of T cells that can also be activated by antigens from this dendritic cell. These are a class of T cells that, when activated, we call helper T cells. Some of these are also going to return to that site where the immune response is and where our killer T cells are going. And again, we'll look at what they do. But as we can see, some of these helper T cells are going to assist B cells. So let's see what's going on there. Like how we said that dendritic cell with its specific piece of an antigen from a specific organism or part of an organism scoured through all of these T cells looking for those that could recognize that specific antigen, we also have scores of B cells, each that can recognize some specific antigen. However, unlike our T cells, which needed the dendritic cell to show it that antigen, B cells can usually see an antigen on their own. They just have to wait until there's enough antigen that's coming from either the vaccination site or the site of infection that actually gets into our circulation and travels into this lymph node. Once it does, though, it will eventually bump up into these B cells, which have antibodies stuck into their surface, and this is how it's going to recognize them. As soon as one of these antibodies on one of these B cells binds or recognizes an antigen, this B cell will immediately start to make some antibodies. However, kind of like your first YouTube video, they won't be the greatest. Thankfully, we have these helper T cells that are going to speed things along. When these B cells recognize this antigen, in addition to making some kind of crappy antibodies, they're also going to pull them in like the dendritic cell did and break them apart so that they can then show off these smaller antigens looking for a T cell that can recognize it. If they're able to find a T cell that recognizes this fragment, then it will be similar to falling in love over the same terribly great movie. The T cell now is going to produce these small molecules called cytokines, and these are going to help the B cell improve its antibodies, kind of like hopefully your friends give you some critiques on how to improve your YouTube videos. With the help of the cytokines from this T cell, this B cell now is able to make much better and much more antibodies. So one last tidbit before we jump back into our site of the immune response and we see how all of that action unfolds. The point of a vaccine and the hope of an infection is that the immune system will develop this long-term immunity. That comes in the form of what we call memory, and it occurs in the lymph node. Looking at our dendritic cell, in addition to activating some T cells to become killer cells and other T cells to become helper cells, it's also going to produce these cytokines, similar to what we saw in the activation of B cells, and it's going to act on some of these T cells. And instead of becoming outright killer or helper T cells to function right away, these T cells are going to become memory T cells. And they're going to hide out in different parts of the body, waiting to see this antigen again. Similarly, we're going to want memory B cells so that if we see this antigen from this pathogen again, we can rapidly make antibodies. The way that we make memory B cells actually relies on those helper T cells, producing some cytokines that instead of improving the antibodies is going to gear these B cells to become these memory B cells, and they're usually going to hide out in the bone marrow. Alright, now that we've covered that, let's see it all together. So with the activation now of our T and B cells, let's see what this immune response looks like. Our activated T helper cells are going to produce more cytokines that will help natural killer cells better kill infected cells and will assist macrophages in their job of destroying pathogens as well as clearing antigens from these pathogens and from destroyed cells. Meanwhile, our T killer cells are going to produce different cytokines and other molecules to help natural killer cells destroy infected and damaged cells. Meanwhile, our B cells are going to be pumping out those antibodies to latch on to those antigens they're specific for to neutralize those threats. Once this threat is taken care of, whether it's just an antigen or whether it is a pathogen, these immune cells are going to reside, and a lot of them are actually going to die shortly after this encounter. But don't forget, we made those long-term memory cells, both B and T, that are going to be sticking around in case we ever see this antigen again. Our immune system is a busy network of cooperating cells. Many of these tasks are occurring simultaneously in different parts of the body, and some require even more strategy because they're going to take days or even weeks, like activating those B cells and making those high-quality antibodies. Also, this is a big picture. We've skipped over and looked past a lot of other players and smaller interactions and molecules that are at work here. More importantly, this is during a vaccine, when we've made the job easy on the immune system. During an actual infection, all of this has to occur while the immune system also has to battle an active infection. And that pathogen plays dirty. It'll use every trick it can to slow down or subvert our immune response. Vaccines then are a really important tool to help us outsmart many of these pathogens. It's like giving our immune system a dress rehearsal to know what to do and how to perform before the night of the big show. So the next time you go to get your poke in the arm, just think about the incredible strategic adventure millions of your cells are about to embark upon to keep you safe. Science really is amazing. If you've enjoyed exploring this topic with me today, like and subscribe so that you can join me the next time we talk about more awesome science.